Okay. Hello, good evening, everyone. As Toby said, my name is Ali, and the title of my talk today is Antibiotic Resistance, the Issue That Will Never Go Viral. And I kind of chose this title for its double meaning. One, because antibiotics don't work against viral infections, so they will never go viral in that way, but also because antibiotic resistance is a massive issue that's not talked about nearly enough in the media and online. And so in that way, it hasn't gone viral. But if we are to fix this issue, it's important that everyone is aware of it. I'd also just like to talk about this picture that I have on here for a second, because Toby didn't realize this, but it's actually a picture I took in the lab of my bacteria. They glow under a fluorescent UV light, which is pretty neat. So I'm going to get started by telling you a bit about me and my journey to grad school. So I'm actually not from Manitoba. I'm originally from Nova Scotia, and I did my Bachelor of Science in Medical Sciences with honors at Dalhousie University in Halifax, Nova Scotia. And through my degree, I really wanted to go to medical school for the first 2 years, but things did eventually change as I gained more experiences and learned about more careers. However, things have changed again, and I do want to go to med school again, and I'm waiting to hear on results as we speak. And so through my degree, I was able to take a lot of really neat courses in a number of different areas. I had courses in anatomy, physiology, neuroscience. That one wasn't for me, ew. And I even took a course in rock and roll. But out of all of these courses, what I really liked the most was microbiology. I was also able to gain a lot of research experience throughout my degree. I had a summer research award where I worked in a sea monkey lab at the end of my second year. I did an honors research project in microbiology at the Canadian Centre for Vaccinology. And this is not something that I did, but I have a lot of friends who were able to gain research experience through co-op terms, and they really enjoyed that and had great experiences. In addition to this, I also had a lot of experience in teaching in science and communication. So I was a lab teaching assistant in cell biology, and I was also a science summer camp instructor for 2 years, where I taught youth from grade primary all the way through grade 12 about science. And so I really enjoyed teaching and communicating science with others and was interested in research as well, which is why I decided to pursue my master's in medical microbiology and infectious diseases, all the way here in Manitoba. And I started in fall of 2019 with my supervisors, Drs. Denise Bay and Dr. George Goulding. And my lab studies antimicrobial resistance. And that was really one of the reasons that I wanted to come here and study in this program, because antimicrobial resistance is a really big deal. So I'm going to take a little bit of a step back to kind of make sure we're on the same page about antimicrobial resistance before we get really into this. So as you probably know, antibiotics are drugs that we can take to cure infections that are caused by living organisms, such as bacteria and yeast. I'm sure many of you have taken antibiotics for things such as penicillin or amoxicillin for infections like strep throat, or maybe even an ear infection. But antibiotics aren't the only thing that we have in our world to keep us safe against microbes. Antibiotics actually fit into a much larger class of things known as antimicrobials. And antimicrobials are just things that are able to kill or inhibit the growth of bacteria, yeast, and viruses as well. So there's many things, and I'm sure you're familiar with a lot of them. This one we've been seeing a whole lot of these last 2 years. Hand sanitizer. There are antivirals, which work against infections caused by influenza and other viruses. If there's anything that you take from this presentation today, I will say it again. Antibiotics don't work against bacterial infections, so please don't take them if you do have a viral infection, such as COVID. In our world, there are a number of naturally occurring antimicrobials that come from plants and plant extracts. So we know historically that these types of antimicrobials have been used often, especially by in indigenous medicines. Next is certain metals, which can be antimicrobial, such as copper. This is actually what my project is on, but we won't be getting into the specifics of that today. Here I have pictures of copper being used on a toilet, where it's often used on touch surfaces to prevent bacterial contamination. but it can also be used in textiles such as masks. I can't really endorse this. I don't know if it's good or not, but it was just something I wanted to show. And then finally, another type of antimicrobial would be disinfectants. So these are types of antimicrobials we apply to surfaces, and they don't go within the body. And these are to prevent or remove antimicrobial that are growing on surfaces. They're also often used within products, and this is where quacks, which are a type of disinfectant, are often used. So they can be used in small amounts within daily household products such as shampoo, makeup, and laundry detergent. And this quacks are primarily what the Bay lab that I work in studies. So where in general around the world are antimicrobials used? So of course, we know in healthcare to treat patients and to ensure that things are sterile and cleanly. Very often and most often in animal farming is where we see a lot of antimicrobials used, and actually 78% of total antimicrobial usage in 2016 was in animal farming. And this is for a variety of reasons, for prophylactic treatment of animals to ensure they don't get sick, or if there is an infection within a herd of animals. In food processing plants to ensure that people aren't getting sick in the process of food being made and delivered to grocery stores, as well as I said before, in a variety of household products. So these are just a couple examples, and essentially antimicrobials are used everywhere. So we know that antimicrobials, if we give enough of these to bacteria or to other organisms, they should die. But with the case of things like disinfectants, where we're adding them in small amounts to products, or in cases where maybe someone doesn't take their full course of antibiotics, bacteria might get a very limited exposure to these antimicrobials. So what happens when they get this small taste? Well, if they're just getting a small taste of antimicrobial and not enough to actually kill them, the bacteria can evolve to try to live better with these antimicrobials. So they're no longer going to be affected by it as much. It's kind of like if you have a very annoying sibling and they just won't stop talking, it's not enough to like push you over the edge, but you do eventually learn to just put a headphone in and block them out. So essentially what happens when the bacteria learn how to live better with these antimicrobials is we call them superbugs. And superbugs are resistant to antimicrobials and they become much harder to treat. And so how exactly does this process of a bacteria evolving to live better with antimicrobials happen? Well, there are two really main ways that this happens within the cell. So there can be genomic mutations. So the DNA within a bacterial cell can have some type of mutation in its code that makes it better equipped to handle these antimicrobials. Or in some cases, there's something known as horizontal gene transfer, which is when bacteria will share specific genes, typically on plasmids, with one another. And these genes are often for antimicrobial resistance because it's helpful for them if the whole population is able to survive. And now some general ways that bacteria can be resistant to antimicrobials. Number one, they might learn, or their genomic mutations or other genes that they have may allow them to limit uptake of antimicrobials in the cell. And they can do this by modifying their bacterial membranes, so making them maybe really thick or hard to pass through. Sometimes they can get through, but can be quickly shuttled out by the bacteria through things called efflux pumps. The bacteria may make a specific type of enzyme that's able to chop up the antimicrobial and make it not work and inactivate it. And finally, they can modify the target that the antimicrobial acts on so that the antimicrobial can no longer bind, but it's still an important piece within the cell that is still functioning for them.
Ali, there's a question from
Yeah.
Good question. So for multi-drug resistant bacteriums like TB, do you know if we're still trying to find new methods to treat them, or are we sort of out of options with our current medical knowledge?
Yeah, so there are a lot of strains of extensively drug-resistant tuberculosis. We are still trying to work on ways to try to solve this issue because most TB is incredibly hard to treat now. But we will get to towards the end of the presentation why it is incredibly difficult to discover and bring to market new treatments for resistant infections like TB. So that was a very good question. You're on the right track. Okay, I'm moving. Any more questions in the chat?
That's it so far.
Okay. All right. So we look at antimicrobial resistance in our lab. What kind of experiments do we do to study this? So first of all, we can do adaptation experiments. So this is essentially simulating small exposures to antimicrobials over a period of time to see what the bacteria do. So we grow them with a tiny bit of antimicrobial, and then we will give them a little bit more and a little bit more. And we do this over a number of generations, say 40 generations of this bacteria growing. And then over time, when we're giving them a little bit more each time in higher concentrations, the bacteria become resistant to higher concentrations of the antimicrobial. And then we can see how they begin to act and different changes they make. So some ways we can look at these changes, we might look at minimum inhibitory concentrations, or MICs, where we grow the bacteria with increasing concentrations of antimicrobials, the one they're exposed to or others, in order to see if they'll survive. And we can also do omics type experiments. So genomics, looking at DNA, transcriptomics at RNA, and proteomics, looking at proteins. So just as an example, for genomics, we might look at all of the DNA within the adapted and wild type bacteria, and then look at what genes that they have gained or made changes to in order to make them resistant. And then to confirm this, we may remove these genes and see how important they are for survival in the presence of this antimicrobial. So yes, we do this research, but why exactly do we care about it? Well, there was a report put out in 2019 called When Antibiotics Fail, and this report really highlights why we need to care about this issue. And the majority of the content from here forward is kind of summarized from this report. So I'll first go through some facts about antimicrobial resistance, or AMR, in a Canadian context. So this report compared 2018 to 2050. In 2018, 26% of infections were resistant to drugs that usually were used to treat them. So that's about a quarter. But by 2050, if we continue on the path we're on, where antimicrobial resistance is increasing, that could go up to 40% of infections being resistant to antimicrobials. In 2018, there were about 5,400 deaths directly attributable to AMR. There are some that were related to AMR that brings this number even higher, but this is directly. And by 2050, this number could go up to 13,700. And for some perspective, COVID-19 in 2020 and in 2021 caused about 14,000 deaths in Canada each year. So this is a quite equivalent problem. And we saw the issues that this caused in our healthcare system, and especially on the economy of our healthcare system. So in 2018, antimicrobial resistance caused about $1.4 billion in cost to the healthcare system. But by 2050, this number could rise to $7.6 billion, which is equal to about a third of Manitoba's GDP, I believe. So that's a 542% increase in cost to the healthcare system. And unfortunately, this gets even scarier when we think about worldwide, because it's expected that by 2050, there might be up to 10 million deaths per year worldwide caused by antimicrobial resistance. That is deadlier than cancer. And just to push this even further, these numbers were based on a study from 2019. So this is probably even worse than we're expecting, and likely an underestimate. And this is because the COVID-19 pandemic has drastically increased our usage of antimicrobials. With people getting sick and going to the hospital, they're more likely to get hospital-acquired infections. We're using hand sanitizer and antimicrobials on everything. So this usage is bound to increase antimicrobial resistance across the world. And something else we know about this issue is that the deaths across the world and within specific populations, even in Canada, won't be evenly distributed. And that's because there are a number of social risks that can make one more prone to acquiring an antimicrobial resistant infection. And the risk of acquiring these infections is not uniform. And we can really see that a number of groups that are vulnerable to antimicrobial resistant infections are once again very similar to those vulnerable to COVID as well. So healthcare workers who are in direct contact with individuals who have these infections, immunocompromised individuals who may not be able to, or who may not have the immune system to properly defend against these pathogens when they enter their body. Age is another factor that may impact one's ability to have an antimicrobial resistant infection, because neonates, which are babies, and elderly people have less developed or not as good immune systems as the rest of the population. And next would be people who provide services. So hospitality, transportation, education, likely grocery store workers as well. And then finally, marginalized groups. So Indigenous people, people living in poverty, the unhoused, prison populations, and people who use drugs. And what connects these marginalized groups in many cases is not just that they have a limited access to healthcare, but very often they have inadequate housing that's crowded with poor insulation and ventilation, or even they have a lack of housing. And these are conditions that allow for antimicrobial resistant pathogens to thrive. I probably could have given an entire presentation today on how colonization has led to poor health outcomes in Indigenous people within the North, which has in turn been caused by poor housing in these situations. And this has led to extensively drug-resistant tuberculosis thriving in these populations. And unfortunately, we're just not doing enough to address not just the infection, but the social causes of this as well. And then hopefully in our workshops, your groups will have a chance to discuss some of the other social impacts of antimicrobial resistant infections as well. So what do we have to do? How are we going to mitigate this problem? So the report has suggested 4 main pillars, starting with surveillance of antimicrobial resistance. It's important that we are collecting data on antimicrobial usage and AMR in various sectors, not just within healthcare, and that this data is very comprehensive and easily accessible for us to be able to survey it and do research on. Next would be infection prevention and control, or IPC. It's important that as we go forward, we are meeting current standards of personal and environmental hygiene in hospital settings, as well as in community care settings. And that in order to implement this, we're providing staff with adequate training and resources to be able to ensure that we're following infection prevention and control methods. And it's also important that we're looking at infection prevention and control across all sectors, including agriculture. And then the next pillar is research and innovation. So you might be thinking, you know what I'm going to say next. We just need to make new antimicrobials. And so as I was saying earlier, it's actually a lot more complicated than that. So there actually haven't been any new antimicrobial classes discovered since 1984. And why is that? Why haven't we just made new ones? And that is because the pipeline from discovering an antimicrobial and going through preclinical and clinical trials, which typically have 3 phases, jumping through all of the hoops for that, and then getting the drug approved and marketed, having facilities produce the drug and then distributing it is incredibly long. This process can take decades, and because it takes so long, and because science just in general is very expensive, it takes a lot of money. And we obviously know that drug companies will focus on making drugs because they do tend to make a lot of money on them, but antimicrobials tend to be an exception. This is because once the drug is distributed, they don't have very much time to make a return on their investment before generic companies can make the drug at a cheaper price. As well, antibiotics are not considered a drug that's used for chronic conditions. So say, for example, with a cardiovascular medicine, once someone begins taking something like that, they're generally taking it for the rest of their life. Whereas with antibiotics, we're just taking them for a short period. So you're not getting as much return on investment. And so overall, the production of new antibiotics is a very high risk, low return scenario. So that is why we really haven't had much development of new types of antimicrobials throughout the last 30 years or so. So what are some ways that we can actually focus on research and innovation to solve this problem? It's important that there are more flexible regulations that make it easier for companies to develop a drug while also still ensuring that the drug is safe. That companies have economic incentives that are supported by the government. So types of funding that can allow them to develop these drugs and still be able to run their business. And also switching our research focus away from just looking at broad spectrum antimicrobials. So we might look at unconventional targets within a cell. Vaccines can be developed against various bacterial infections. Phage therapy, which phages are viruses that infect bacteria. And we can take what's called a cocktail of phages and give them to a patient. And finally, adjuvants that can increase the efficacy of antimicrobials. And then the last pillar is antimicrobial stewardship. So this is kind of the idea of taking action and making change to our current antimicrobial habits as they currently are. And the article mentions doing this through a One Health approach. So One Health is essentially this idea that we have to collaborate between human, agriculture, and environmental sectors in order to tackle a variety of issues, but in this case, antimicrobial resistance. So for example, antimicrobial stewardship may involve appropriate prescribing of antibiotics by practitioners, both doctors and veterinarians, appropriate antibiotic usage by patients, so only taking antibiotics when you need them and taking your full course of antibiotics, and not kind of tying into the last one, not asking your physician for an antibiotic if it's against their personal judgment. Reducing unnecessary antimicrobial use. So that can be in all of these sectors through implementation of policy. So for example, antibiotics are often used to promote growth in farm animals. So a number of countries have banned this in agriculture in their countries in order to reduce antimicrobial usage. And finally, education is incredibly important for this as well. So this involves ensuring that everyone, healthcare practitioners, farmers, the general public are aware of this issue and are aware of what they can do to solve this issue. So hopefully I was able to give you some of that today. So that is all for my presentation. And I'm just finishing off with some of my favorite AMR related memes. And I guess we can take a couple minutes for questions.